Welcome to part 3 of basic concepts in immunology. So just keep this here, making sure that we are um, continuing our discussion on some of the main parts of the immune system. So the responses by the immune system may be classified as non-specific responses or adaptive responses. So let's say a few words about each of these. Innate responses provide a first line of defense against the unwanted, against those persona non gratas. Okay, so these innate responses include barriers such as the skin, and this is also something that's going to include something like immune cells fighting pathogens in a non-specific way. Whenever we break on a fever, our body it's also basically following up with one of these innate responses. On the other hand, we have specific responses or adaptive responses. Then here we mean there are cells of the immune system that are going to carry on some receptors. And these receptors will recognize the physical structure of a pathogen. So if you want to make an analogy here with law enforcement, think about some type of like facial recognition software, and I don't know, located in some place, and that's scanning people walking by, and then they are looking to have a match with some reference. Okay, and as soon as there is a match with some specific pattern or structure, then there is a red flag, and this is a similar an analogy or a motivation as to how some of these specific responses are triggered by the immune system. Concretely, uh, the cells in these immune cells will recognize proteins in the surface of the pathogen, and upon recognition, the immune cells would deploy, start dividing and expanding. This divide and expand is basically they clone themselves and for a specific purpose, which is get rid of the pathogen. So they increase in number substantially and enable a fight against the pathogen, and this results in clearance of infection. So let's now talk about a specific virus. The HIV is a type of virus that's called a retrovirus. The particle of HIV, a particle of HIV, would contain two copies of the viral genome in the form of single-stranded RNA. And after after an HIV variant enters the target cell or host cell, then what occurs next is that a reverse transcriptase takes place, and that comes with the virion that actually uses copies of the RNA genome to reproduce, to produce RNA and DNA, and these words here are a little bit of a mouthful. Um, it produces RNA and DNA heteroduplex, and eventually this yields what is called double-stranded viral DNA. Now, this viral DNA, also referred as provirus, is integrated into the genome of the host cell, and this is the part that has to do with hacking the cell. The provirus can remain silent for a long time, or it has also the ability to immediately induce the host cell to produce, and this is an important part, to produce something that's called mRNA, M for messenger. This is messenger RNA. So the viral mRNA has a dual purpose. On one hand, It is used for biosynthesis of viral proteins, and on the other hand, it serves as viral genome for the virions that will eventually leave the target cell or the host cell. So I hope that most of what I just told you is represented on this schematic here. But this is again reinforcing um, some of what we had discussed that has to do with the life cycle of a virus, and HIV is um, one of those pathogens that we are going to revisit with mathematical modeling. So let's talk some more about HIV. And these facts are things that have to do with a profile of this virus, specifically from a clinical point of view. We say that the pattern of disease progression has three phases for HIV. This phase, there is a period of time where it's a short primary phase and is associated with large amounts of virus. Here we see this word appear, or better said, this phrase appears by the first time. Virus load, and this is another word, another way of talking about size or volume of virus. Amount is an amount, how much virus there is. Okay, and the next phase has to do with uh, long and variable phase. This is called an asymptomatic phase, and it's typical of this period to have a lot less virus, fewer, lower amounts of virus load on phase number 2. And then patients go on phase 3. This is where things become rather bad for patients. This phase has a name: acquired immune deficiency syndrome. Okay, so you can think of phase 1, the patient becomes infected. Phase 2, um, there is a lot of variability and there is not many symptoms. Asymptomatic means there is no symptoms. The patient may not even know that is HIV positive. But for sure on phase 3, there are symptoms, and this is why this pandemic of HIV and HIV/AIDS is well known, because this is the part in which the patient basically loses the functionality of his or her immune system. And a simple infection that would not be a major issue for a person that is not infected with HIV, it becomes fatal for an HIV positive and an AIDS patient. Okay, so in the absence of therapy, the average length of phase number 2 may be average, maybe 10 years. During the asymptomatic phase, there is type of cells, they're called CD4. We'll talk more about them later, but um, this this brings another good point here. This is pointing that uh, there is a measurement on the CD4 cells, and that measurement drops below from the order of magnitude 10 to the 3, from 1000 to essentially 0. Okay, so this CD4 and virus load are going to be two things to keep in mind. Okay, given the characteristic time scale of the infection, um, given the characteristic time scale, um, the infection is on the order of days. So then this bears I'm sorry, let me repeat this. If we're dealing with phase one in the order of days, then why does it take years to develop to go from HIV into full-blown AIDS? Okay, this is a scientific question to keep in mind. And we're going to end this part of the discussion with a graphical representation of some of the things that we just mentioned. Here, you want to look at this as a graph where time is in the horizontal axis, and we have one representation for the virus load or the amount of virus or viral load. So we can see how this starts from a low level, it grows up to a peak or maximum level, and then it drops, it remains flat, and then it goes on the rise again. Okay, so this initial peak is within the phase one, within that primary phase. These other levels, um, flat and they're almost not detectable levels, are happening on the asymptomatic phase, and then on phase three, which is known AIDS, this is when the viral load goes on the rise, and the counterpart would be the cells of the immune system that actively are clearing infection. And in phase one, you can see how they lower a little bit, but then they kind of are stimulated by the full amount of large amounts of viral load. But once asymptomatic phase kicks in, then the CD4 counts really comes very low. So we're going to see what we will see next has to do with mathematical models that address some of these dynamic features. And just stay tuned.